So I think we left off on Monday talking about internal things inside of a muscle fiber. We talked about, right, we talked about the sarcoplasmic reticulum, we talked about T-tubules, we talked about mitochondria, we mentioned the nucleus. There's another sort of cellular structure that is going to be in the muscle fiber, okay, that are really cool and they're really, really important. And so they are called satellite cells, all right? What you can see here is this is an individual muscle fiber, and they've sort of stained it or dyed it so that when you take this picture of it, it looks really, really cool. What you will notice is these structures here that are kind of the chartreusey yellow-green here, those are nuclei, okay? Those are nuclei. Remember, muscle cells have many nuclei, and they tend to be out at the edges so we can pack lots of actin and myosin in the middle, okay? Really big, need lots of nuclei. Well, under a regular microscope, satellite cells look like nuclei, okay? But they are not, in fact, nuclei. Satellite cells are going to be in your muscle fibers, and they are important for regeneration and repair of fibers. They're really important for stimulating hypertrophy of muscle fibers, okay? Satellite cells are muscle-specific stem cells, okay? Stem cells. Anybody know what a stem cell is? What stem cells potentially do? That's right, okay? A stem cell, if it is a pluripotent stem cell, can, if given the right instructions, become any kind of cell in your body, okay? That is why there has been lots of talk and controversy around what are we doing with stem cells. Like you can get stem cells from the umbilical cord and things from that kind of fetal tissue from the umbilical cord. And those cells can potentially grow into anything that we need. You need a heart transplant, we'll take one of your stem cells, we'll give it the right input, and it grows a new heart or a new kidney or a new liver, or something like that. Now, we're not to the place that we can totally do that, but that's the potential in those kinds of things, okay? Well, satellite cells are not exactly like that. They can't become any kind of cell, but they can become new muscle cells, okay? And so you're born, you have these, and a lot of them sit, and they just sit in the fiber waiting to be turned on. That means they're what we call quiescent. Okay, and through a host of signals that we're not going to talk about because it's beyond the scope of this class, they can become activated and they begin to proliferate and divide. And they eventually are going to divide and then kind of fuse back together. And they're going to make these things called myotubes, which become myofibers. And they're eventually going to be able to integrate themselves into an existing muscle fiber or to completely form a brand new muscle fiber if one has been totally pulled off. Okay? Anytime you're sore, you've got muscle damage, you have inflammation, but those damaged cells, right, that have caused that immune response, those damaged cells are going to have the satellite cells inside of them most likely activated, and they're going to play a role in repairing. You activate a satellite cell and it divides, but it keeps one of those things, one of these cells, as they begin to split, one of them turns back into a quiescent satellite cell and stays there so that you can have them going forward. Okay. We'll mention these just a little bit more when we talk about muscle hypertrophy because they're really important. Okay. There are certain cellular pathways, there are certain hormones that stimulate these satellite cells to proliferate, and that's really, really important in helping our muscles hypertrophy. We don't do this in humans, but in some animal models, you can zap muscle fibers with radiation and essentially deactivate or kill off their satellite cells. And then you have them do exercise stuff and their muscles don't get any better. So we know that they're really, really important for us. Okay. All right. So Dr. Mike will talk more about those in exercise phys. If you take advanced strength and conditioning, you should talk about them more. I just want to bring them up so that you are, in some ways, mildly aware that these are a thing, that they exist. Okay. Now, let's move down from muscle fiber down to a myofibril, okay, down to the fibril level, all right? So here's our muscle fiber back here. And remember, they're like Russian nesting dolls, tube inside of a tube. So if we pull out one of these individual tubes from in a muscle fiber, it looks like this, okay? So think of a myofibril as a string of soda cans, okay? A string of soda cans. Each can represents an individual sarcomere. So myofibrils are less important than the things that they're made up of, okay? And so a myofibril is a series of these sarcomeres. So it's one, next one, next one. They're lined up in series like this, okay? And so sarcomeres, this is the basic functional unit of a muscle. Whenever you evoke movement, okay? If we ask Kaylee to stand up and do something, you don't have to, Kaylee, all right? We can ask Kaylee to move, and all of that movement is generated by changes at the level of the sarcomere, okay? And so this sarcomere moves, it squeezes in generally. That pulls on the adjacent sarcomere, which pulls on the adjacent sarcomere, which eventually pulls on the connective tissue of the fiber. So the fiber moves, and then the fiber pulls on its connective tissue, which pulls on the fascicle, blah, blah, blah. Eventually we get actual movement, okay? So the sarcomeres where the real action from a movement standpoint and a force standpoint is going to come from, okay? So in Exercise Foods with Dr. Mike, you will learn about these individual kind of zones and lines in the individual sarcomere, right? Places where actin and myosin overlap are called one thing. Place in the middle where there's only actin filaments is called something else, right? There's this place in the middle called the M line, which stands for middle. What I want you to know about the sarcomere, the only thing that I care that you know is the end of the sarcomere, where one sarcomere ends, the next one begins, is called the Z line or the Z disc, okay? And those things are important because the actin and myosin filaments, especially the actin filaments, are anchored, okay? They're anchored or tethered to the Z line. So when you pull on those actin filaments in the crossbridge cycle, you pull them together, okay? They're going to pull on the Z line and move the Z line in, and that pulls on the adjacent sarcomere, okay? So that's why I want you to know that particular structure. All right, so I'll show you guys here this blow-up cartoon of an individual sarcomere to kind of get into the stuff. All right, so sarcomeres are a collection of filaments, or a collection of these sort of smaller tubes that are going to make up our bigger tube that is the sarcomere or the myofibril. Okay. Sarcomeres have primarily two proteins, actin and myosin. Okay, actin and myosin. Actin is going to be a spherical protein, and actin has multiple sort of subunits, but they're put together where you have these winding kind of ropes made up of these individual actin molecules. Okay. So that's what we call a thin filament or an actin filament. Myosin, on the other hand, myosin looks like this, okay? Myosin is a molecule that has a very distinct, what we call a head region, and then a tail region. And so these myosin filaments, what happens at the filament level is that all of the tails get hooked together, and then the heads are going to project off of that at roughly 60-degree angles in space, okay? And so, you know, one of the ways that I think about this is, to me, a myosin molecule kind of looks like a sperm molecule, but its head is sort of tilted up. So the tails of all of them are together, and then the head molecules are going to come up from this, okay? So in our sarcomere, every individual actin molecule has a binding site for the head of myosin. Okay? Every individual one does. There's way more actin, binding sites on actin for myosin than there are actual myosin heads. That's a good thing. Okay? And so if we want to initiate a muscle contraction, what we need to do is tell the myosin head to bind onto that actin filament, and then the myosin head is going to move. It's going to tilt, right? And it's going to tilt on this side of the sarcomere. It's going to tilt in and pull the actin this way. On this side of the sarcomere, it's going to tilt this direction. It's going to pull them in, okay, from right to left. So they're designed to squeeze those actin filaments together and move them into. Okay? Now, here's where the problem comes in. Okay? Here's where the problem comes in. Myosin is really attracted to actin. Okay? It's really attracted in like a creepy way, in a stalkerish way. Okay? If given the opportunity, all myosin wants to do is bind onto the actin molecule at its binding site. Okay? And so, at rest, because of that strong affinity at rest, we have to block the myosin binding site on actin. Okay? Again, I use lots of examples because I have a toddler, but there is a protein that exists on the actin filament that functions like those little clear plastic plugs you put into the wall sockets to keep your toddler from sticking her fingers or a fork or whatever else she can manage to get her hands on, which is everything, into the wall socket. Okay? And so that protein is called tropomyosin. Okay? If you did not have tropomyosin, okay, if you didn't have tropomyosin, every one of you, when you're trying to rest, every muscle in your body would be cramping. Okay? You would exist just like this, just curled up, cramped. Okay? Because the affinity of myosin to actin is so strong that if the binding site isn't covered, it's going to bind, it's going to generate force. Okay? So you couldn't do anything. Evolutionarily, it's better for us to block it at rest and then open up that binding site when we want a muscle contraction. Does that make sense? You guys with me in the midst of all of that? Okay. So it's on there. How do we move it? How do we get it out of the way so if we do want to do a contraction, that can happen? Well, there's a second protein called troponin. Okay? Called troponin. Troponin is a protein that binds calcium. It binds the calcium coming out of the sarcoplasmic reticulum. It has multiple subunits that you'll learn about in exercise phys. But it binds calcium and then it changes its shape. It's called a conformational change. It changes shape. As it moves, it is attached to the tropomyosin. As it moves, it picks it up and it scoots the tropomyosin out of the way, opening up the binding site so that the myosin heads can come in and bind. Okay? Then, very quickly, that calcium is let go of by the troponin, and we break off the myosin head, tropomyosin covers the binding site up again, and voila, you shut off your contraction. All of this happens almost instantaneously. Okay? Let's follow with all of it. Okay? This is going to become really important in a little bit. We're going to walk our way through sort of all of the signaling steps to actually get to a muscle contraction. These are very important principles. Okay? All right. When myosin binds to actin, that forms something that we call a crossbridge. Okay? Crossbridge. Formations of crossbridges allow force production and allow movement. They allow movement. They at least allow the generation of force. Okay? So if I want to generate as much force as I possibly can, I need to, within a given muscle, form as many cross bridges as I possibly can. One of the reasons that increasing the size of your muscles makes you stronger is because you have more actin and myosin, and therefore you can form more crossbridges, thus allowing you. It's like having more people on your tug of war team or something. It will allow you then to generate more force. Okay? All right. Questions about that? Questions about that? Because we're about to talk now about how this gets regulated and how it forms and breaks and reforms and breaks. And that's going to be the really important thing that lets us generate. Okay? We know we're going to talk about fiber types first. Okay. Sometimes I surprise myself on like what order do we do things. Okay. I should cover this. Be like, anybody squeamish about seeing tissue or blood or anything else? McKenna's like, nope, bring it on. You're ready? Okay. All right. So how many of you guys have heard of something called a muscle fiber type? You guys probably talk about this in human phys or in anatomy or something a little bit at least. Yes, no, maybe, sort of, yes, okay. So one of the reasons that we know that there are different kinds of muscle fiber types is because of a technique that was developed in the 1960s. And that technique is called a muscle biopsy. Okay? Specifically, what you all see here, this is what we call a Bergstrom biopsy needle. It was developed in Sweden, I believe. Okay? And so the idea behind all of this is until the 60s, all we knew about muscle, we had to either get from a cadaver or from an animal where we would cut open their muscle and take it out. We did not have a way to learn things about your muscles without you dying. Okay? So the advent of the biopsy, you make a small, like 1 centimeter long incision, usually in the vastus lateralis. Okay? There are not major nerves or major blood vessels out here on the lateral side of your quads, so the lateralis is a good place to go. You cut the skin, cut through the adipose tissue, cut through the connective tissue, open up a window down into the muscles. The lab I worked in for my PhD, we did this. Okay? You make a little small incision, you insert your biopsy needle, which is not quite as big around as a pencil. Okay? It's like 2/3 of a pencil. You insert the needle in, you apply some suction to it, and rather than being a needle in the traditional sense that you think of, where it's open on the very end, it's closed, but it has a little window up a little farther, okay? And so you stick it in and you apply suction and you suck a piece of muscle into this window, then you plunge down on one end, and it's basically 2 needles within each other. And this inside thing is like a guillotine. It's very sharp. The muscle gets sucked in, it comes down, it cuts it off, and you capture that piece in there. You pull it out, and it looks like the eraser on a pencil. Okay, it's about that big, like the eraser on your pencil. And then some poor grad student has to take it and mount it on a piece of what looks like chewing gum on a popsicle stick and dip it in liquid nitrogen, freeze it. And then we take it and you section it using like, what is a deli meat sectioner that gives you like the finest, like little cuts of things that you've ever seen. Okay? So from doing all of this though, we could then begin to learn things about what's going on inside those individual muscle fibers from you. Okay? What happens? How big are those fibers? What's going on with their myosin heads? How much glycogen is in those muscles? How much, what kind of enzymes from different metabolic pathways do you have? Okay? And you can, in theory, do these from pretty much any muscle, but they're mostly going to be taken from the vastus lateralis. Okay? And so from doing all of this, you all probably kind of like you never lived in a world where you didn't have a cell phone or the internet. Okay. You've also never lived in a world without immunohistochemistry. And so in the 60s, one of the other things that began to develop outside of these biopsy things was sort of the advent of some sort of biochemical method. So from this, what we were able to do was take these individual muscle fibers and bathe them in ATP at certain pHs and things and measure how quickly they're myosin ATPase enzymes. So the head of your myosin molecules has an enzyme on it called an ATPase, which is, it means it breaks down ATP. Okay? The myosin ATPase, as you guys will see, is one of the key regulators of muscle contraction and muscle contraction speed. Okay? And so what we learned is that based upon the speed of the myosin ATPase, your muscle fibers exist in roughly two kinds, what we call fast and slow. Fast fibers do just what you think. They break down that ATP, that enzyme works fast. The slower ones, it works somewhat slower, okay? And so we originally thought, oh, look, there's two kinds. There's fast fibers and there's slow fibers, which was more than we knew initially, okay? But look, they're not all the same. It's kind of cool, all right? And then as we moved forward and we got even better at doing biochemistry, we were able to develop these very specific monoclonal antibodies to not just the enzyme, but to another piece of the myosin head called the myosin heavy chain. Don't worry about what the heavy chain is, or there's two heavy chains and two light. Don't worry about those things. Dr. Mike will cover all of that in exercise phys. But what we learned from that is that the ATPase enzyme is fast and slow, but there's also another piece of the myosin that is a little bit different among fibers. And so from that, we were able to figure out that slow muscle fibers have one, the MHC stands for myosin heavy chain, one isoform of this myosin heavy chain, one flavor, okay? And so we call those type 1. I will use type 1 or slow twitch fibers interchangeably. Okay? It means the same thing. Okay? But we learned that fast fibers or fast twitch fibers have multiple kinds of myosin heavy chain isoforms that they might express. So there's differences among the fast fibers. Okay? In general, okay? So we call these, the fast fibers are called type 2. They exist in the flavor of either type 2A or type 2X. Okay? A or X. Type 2A fibers are metabolically a little bit different than type 2X fibers. Their myosin ATPase speed is roughly the same, okay? But type 2A fibers have the capacity to be much more metabolically efficient than type 2X's. They can have a lot more mitochondria. They can have a lot more capillaries and things that are going to go to them, okay? So we can also fiber type muscles based upon their metabolic activity, right? Sometimes you guys may have learned them this way, even though it pains me to say all of this in human phys as slow oxidative fibers or fast oxidative or fast glycolytic fibers. Does that sound like something you guys might have heard of previously? Okay. I realized this that one of my master students works in as a tutor for, I think it's for human phys. She was asking about that, and I was like, no, please don't call them that. Like it's really, in some ways, kind of not the most correct thing. So briefly, indulge me, okay, indulge the graduate student in me. All of these fibers can do oxidative or aerobic metabolism and glycolytic metabolism. When I say something is a slow oxidative fiber, it is also a glycolytic fiber, but it just predominantly can make most of its energy oxidative. The type 2X fibers are predominantly glycolytic, okay? Just because they don't have very many mitochondria and they're wildly inefficient. So they do oxidative metabolism, but just not very well or not for very long. And so in that way, I think these are slight misnomers in the way that things are today. But type 1 fibers or slow twitch fibers are the most metabolically efficient, okay? To be a world-class, elite, long-distance runner, you probably have lots and lots and lots of type 1 fibers, okay? Type 2A fibers are less oxidative, okay? Less oxidative than type 1s, but they're more oxidative than type 2X's. if you train and you run or bike or elliptical or row a bunch, I can take 2A fibers and get a lot of mitochondria in them and be very good as a distance athlete doing all of that. Okay? And so people that are maybe, you know, elite kind of 800 meter or mile or even 3,000 meter folks can get away with having more of these 2As, okay, being a little closer to 50-50 here with the 2As, right? Because the 2As let them actually be faster. They can physically run faster than if you have all type 1s. 2Xs suck. 2Xs are like a Hummer or something, okay? You get like 3 miles to the gallon. Contract them twice and they're tired, okay? Most of you have very, 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 very few 2X fibers. If you move at all, your 2X fibers will turn into 2A fibers. And then if you're a couch potato for a while, your A's will turn back into X's. 2s to 1s doesn't happen. 1 to 2s, if you have a spinal cord injury or you're bedridden for years, maybe we can get some 1s. But in general, your fiber type comes from your parents. You cannot change this, okay? You cannot change this. Almost all of you in class, probably with a couple of notable exceptions, are 60-40 one way or the other, okay? You're 60% type 1, 40% type 2. Or you're 40% type 1, 60% type 2. Most people are somewhere in that range, okay? Or, you know, 70% of people are going to be within that range. Within your muscles, okay, most of your muscles are going to be very similar in their fiber type distributions. There are some notable exceptions. Your diaphragm, mostly type 1, okay? You've been contracting your diaphragm for pretty much your entire life. It mostly doesn't get tired, does it? Can you imagine doing, you know, like squats every time you took a breath for your whole life, how tired your legs would be? Okay? Your diaphragm doesn't. Postural muscles like your soleus, okay? Some of your lower back muscles and things, they're going to be a little more. If you're normally a 50-50 fast to slow, your soleus might be 60-40 or 65-35, somewhere there. Okay? But that's where we are. All right? Whatever you've got, you've got. If you're a good distance person, thank your parents. If you're a good sprinter, thank your parents. If you're like the rest of us, you're like parents, what the hell were you doing? Have better genes. Come on. All right? Any questions about this? No? Sure? Can you envision an essay question on the test whereby I say, compare and contrast the muscle fiber types to each other? Talk to me about contraction speed. Talk about their metabolic activity. Talk about what's happening at the myosin ATPase or something. Okay, so here's some things that are going to kind of get at a lot of these things that are going to be differences in the fiber types. You guys can look at all of this. It's relatively straightforward. Okay, type 1 fibers generally are going to have all of the things that we need to be good at endurance. 2As are somewhere in the middle, and 2Xs are not good for endurance, but they're really good for lots of force and sprinting. Okay? All right. Let's move away from muscles then. Okay? Talk about the nervous system. I asked Adriana to stand up and touch her nose. Can you do that? Yes, please. Adriana, stand up, touch your nose. Okay? Now, put your left arm out. Yeah, don't hit, fail. Okay? Now, hop on one foot. Very good. Okay, you may sit down. Thank you, Adriana. What she just did was conscious control. I asked her to do something. She thought about how to do it, and then she did it. Right? All of that is driven by the nervous system being able to tell muscles what to do. Right? We're not going to get into the neuroscience and the neuroanatomy of the brain and the motor cortex, the primary cortex, secondary cortex. and kind of the higher order things in the brainstem. We're not going to talk about that. Right? There's some great classes in biology and neuroscience and neuromuscular control. If you want to take those, I highly encourage. Okay? But if we want to initiate a muscle contraction, we're going to assume that your brain and the motor cortex functions properly and that you've got reasonable transmission down the spinal cord. Where the big things start for us in the nervous system is at the motor neuron. Okay? The motor neuron is a specialized nerve cell. That nerve cell is in your spinal cord. Okay? It's in your spinal cord. We're going to learn a little bit about its anatomy, okay, and the structure of motor neurons. We're going to learn a little bit about where in the spinal cord they occur spatially. Okay? They're very specific. And then we're going to talk about how they send and receive signals. They're going to receive signals from the central nervous system and from the periphery. And then they're going to send signals out to your muscles to tell them to turn on or turn off. Okay? Here's a cartoon of a motor neuron. Okay? All neurons or all motor neurons have a cell body which sits in the spinal cord. Okay? Surrounding that cell body are these little thin projections that are called dendrites. Okay? Dendrites are going to help pick up electrical signals from all around the area. They're going to give that information to the cell body. Okay? Then this part sits in your spinal cord. Okay? So out here at the very end is going to be muscle fibers. So we need a way to communicate from our cell body in the spinal cord out to your individual muscles. That is done via this very long structure here called an axon. Okay? Every motor neuron has one axon or one primary axon that branches off of it that comes out of the spinal cord, okay, through these sort of large nerve bundles that exit via the foraminal space. They come out. They're surrounded and wrapped in something called myelin, which is a fatty sheath that increases conduction velocity so we can send a signal very, very fast. The axon comes out, and then it travels all the way down out into your muscles. Then it begins to branch into these little individual sections that are going to come into close contact with individual muscle fibers. And so in that way, signal comes from the brain down, picked up in the dendrites, tells the cell body to polarize, shoots that action potential down the axon, and then that's going to come out here and be communicated to our individual muscle fiber. Okay? Axons are huge, very, very long. Right? Very long. Imagine, right? The motor neurons that are going to go to the muscles that control my ability to move my feet or my toes sit at about right here on my back, at about somewhere between S1 and S2. Okay? I have to have an axon that runs from here all the way down my leg, down to those individual muscles. And it has to be able to transmit an action potential just like that. I put my foot on something hot or something very cold or something sharp, and I got to move it out of the way. Okay? Information has to travel up and then back down like that, so I don't get significant tissue damage. Okay? And so long things, but action potentials, electrical signals travel very fast. We have this myelin that lets that happen. We can communicate very, very quickly. Okay? In some very large animals, okay? Giant squid, for example, have like some of the largest or the longest axons in the world. They have some of their, what do you call it? I'm going to call them a tentacle. I don't know if one of their arm things that are super long are like 60 feet long. Okay? They have an axon that runs from their, basically from their brain, because they don't really have a spinal cord because they're squid, all the way down. Okay? Huge, long, long thing. You guys saw nerves, or what we think of as a nerve is really a nerve bundle, right? You ever bump your funny bone, you bump the nerves, you bump your ulnar collateral nerve or something here. You guys saw those nerves or those nerve bundles in anatomy class, I would imagine. They're really tiny, right? I'm always astounded, like, how do we not like fall over and just like rip all of our nerves in half all the time? What you're seeing are those nerve bundles are actually a bunch of axons all together. A bunch of axons. Now something that's a little bit more complicated, but really, really important. Okay? Really important. Every motor neuron will connect to many muscle fibers. Please write that down. Every motor neuron will connect to many muscle fibers. That collection of structures. So the motor neuron, all the fibers it connects to, that is called a motor unit. Right? Motor unit. All of the fibers that are connected to one motor neuron are going to behave similarly. Because what happens is, when this motor neuron gets turned on, it will tell all of the muscle fibers to which it connects to turn on and contract. Right? So they function as a group, basically. Now, every neuron connects to many fibers. Every muscle fiber in your body is connected to a motor neuron, but it's only connected to one motor neuron. Okay? Only to one. So there's not going to be any kind of crosstalk. There's not going to be any kind of confusion. Right? Within a given muscle, you may have several hundred to several thousand motor neurons that connect to fibers in that muscle. Bigger muscles tend to have more motor neurons. Right? Within that pool of motor neurons that all connect to that muscle, there's going to be large and medium-sized and small motor units. You're going to have some motor neurons that connect to a small number of fibers. You're going to have some motor neurons that connect to a huge number of muscle fibers. And there are plenty of them that are going to exist in the field. Right? We're going to talk about the importance of this setup probably next Monday. Right? But this arrangement, the way that this structure is arranged, is of the utmost importance for our ability to generate very precise amounts of force. Okay? If it wasn't set up like this, we couldn't do the things that we do. Okay? We'll try to talk more about that as we go along. The other piece to keep in mind with all of this, okay? All of the muscle fibers that are connected to a motor neuron, all of those fibers are the same fiber type. So all of these fibers here have the same fiber type. All of these in this larger motor unit, all of these have the same fiber type. And there's a pattern to the size of the motor unit with the fiber type of the fibers in that unit. Okay? Smaller motor units, the ones that have the fewest number of fibers, those tend to be type 1 or slow twitch fibers almost exclusively. The biggest ones are 2X fibers. And the 2A's are somewhere in there. Okay? Again, this arrangement is really important for how things function. It's really important. We'll talk more about that, the why. Okay? One of the things that we know that it's, to me, super fascinating. Let's assume this, okay, is what these are slow twitch fibers. So we would call this a slow or a type 1 motor unit. This is a big one. This is a type 2X motor unit. They have done experiments, not in people, but in animals, where they go in and they cut this axon and they cut this axon and they swap them. They reconnect them, but they swap them around. So if you connect these fibers to this neuron, they stop being type 1 and they switch and they become 2X. And these stop being type 2X and they switch and they become type 1. So while genetically your parents determine your fiber type, we think there's a lot of things what those things are is driven by the motor neuron as much as it is by the actual fiber. There's some really interesting stuff involved. Okay? All right. Let's stop here. We'll take a little break, okay? And then we'll reconvene.
Professor, your mic's not on. Professor Black, your mic. Your mic's muted. I don't know if you can hear me.
All right, Zoomers, can y'all hear me now?
Yes.
Okay, Jordan says yes. Okay, we're back. We're live. Sorry. Technical glitch. Y'all missed, Zoomers, the single most important thing that's ever been said in class. Except you didn't. All right? So all of the motor neurons for a muscle are in the same place in the spinal cord. Why would that be good? Good about it. Devin, what do you think? It would be good about having all of the motor neurons for a given muscle in the same place.
hurt in like one area, it's not like there's not, it usually just affects like one area, so it's affecting certain body parts instead of like
that's right.
being spread out.
Sure. That's definitely a downside of those things, right? That was going to be my next question. So that would answer this, my second question very, very well, right? The downside of this is if you damage the spinal cord or you get an injury somewhere, then you're knocking out kind of everything below, you're knocking out all of one particular muscle. I guess you could argue that if you had some kind of an acute thing at like C5 or something, then you're just going to lose that muscle and not a bunch of kind of other ones. That's potentially good, okay? What else? What could happen though if I had some deltoid muscle here, I had some down in the thoracic region, I had some in lumbar, and I had some down in the sacral area as well. They were really, really spread out. Lauren?
It can be hard to communicate in the nervous system, and signals can get crossed easier.
Okay. Harder to communicate, right? So I am not a particularly tall person, okay? But there's still distance from here down to here, right? And if for some reason I needed to turn on all of my deltoid muscles simultaneously, like I'm having to, you know, I don't know, like military press a car or something up over my head for some, you know, who knows? You never know what happens in life, okay? Then if I've got some here, they're going to get turned on first. And then it takes, you know, fractions of a second, but it's still some delay to get these turned on down here. And then even longer to get these turned on down there. And so that, you know, that could be a problem, right? That could be a problem. So there's some adaptive advantages. They're all in the same place. It's easy to get them all turned on relatively simultaneously and quickly if you need to, and turn them off in the same kind of way. All right, but then the problem with that is when you get damage to the cord, you can knock out all of them. You can knock out everything below that particular level. Right? So just some things to keep in mind. Those of you that want to be neurologists, or you want to be a physical therapist or an occupational therapist, we can learn a lot about where, if there is a spinal issue, if there is impingement or inflammation or something going on, we can learn a lot by looking at individual sort of sensations and strength and things in individual muscles. And we can track that back to these very distinct locations in the spinal cord, which can then tell us, look at this place. Is there some anatomical problem there? Usually there is. We'll get into sort of low back issues at the very end of class, and we'll talk about what happens when you have, let's just say, herniated disc or something, which of these things are the most. You get these very, very distinct presenting pathologies. I have pain in this area. I have muscle weakness in this area. And it's because all of those motor neurons and all of those sensory neurons are in these very specific places. Very, very, very specific. Okay. All right. Okay. I think I mentioned this. We're going to do it formally. The nervous system communicates most of the time. Okay? Let me rephrase that. The somatic nervous system that controls motor function generally communicates with an electrical signal that is called an action potential. Right? An action potential. So an action potential is a brief electrical signal. It moves from one place to another. That's often called propagating, or the signal is propagated, okay? And it occurs because of a change in membrane potential. So membrane potential just very simply means, right, this is a nerve, okay? And based upon some collection of ions, usually the inside of the cell is more negative than the outside of the cell or something like that. And there's this difference in electrical charge across those. And what happens then is to get an action potential, I open some channels that are going to be on here, and it allows these charges or these ions that have these charges to move back and forth. And that sort of moves its way down my nerve in that particular way. Okay? Again, we're not going to spend a huge amount of time talking about action potentials. I'm sure you guys covered that in human phys. All that we need to know is that's how they communicate, okay? Electricity is fast, which is good, right? Electricity is fast. There are some problems because then as we change membrane potential, there can sometimes be a delay before I can send another one. Okay? Action potential in the motor cortex, in theory, is the first step to a muscle contraction. Okay? You've got action potentials, motor cortex, down the spinal cord. Action potential causes the motor neuron to generate its own action potential. It moves down the axon, so on and so forth, until it reaches the muscle. Okay? In theory, if an action potential is prevented by a drug, muscle contractions will not be possible. Okay? You guys ever been to the dentist when they do, like, you have a filling done or something? They inject you with Novocaine or something in your mouth. Or you've had any local anesthetic and they're going to do some kind of thing, they inject you with xylocaine or something like that. Anesthetics block action potentials. They block the ability of those things to be generated. A lot of local anesthetic, it blocks both the sensory and the motor function. Very non-specific, although we can sometimes do things that are very specific to one or the other. And so you cannot contract the muscle. That's why you get out of the dentist and your face is kind of sagging, right? And all of that. It's because we block the ability to generate action potential. Okay. So, action potential, it's moved, it's come from the motor cortex, down the spinal cord, into the motor neuron, down the axon, gone down the big axon, it's out into the muscle, and it's going to go out to these little branches we call axon terminals. Okay? Axon terminals are going to come down and get very close, but not quite make contact with individual muscle fibers. Okay? This area where they get very close is called the neuromuscular junction. Nerve muscle junction. Okay. So this is kind of a big picture look at all of this. The neuromuscular junction is a very highly specialized structure. And on, kind of a very small level, it looks like this. Okay. The ends of the axon terminal are going to spread or kind of bulge out. Okay. And then on the sarcolemma, right under that area, it's going to fold. Okay. It's going to fold. Both of those things increase surface area. We have a larger surface area on the nerve side and a larger surface area under that nerve on the sarcolemma. This helps us by having more surface area to ensure that we can release a small amount of a neurotransmitter called acetylcholine into what we call this area here, okay? Right under it is called the primary neuromuscular cleft. The areas down here in the folds is called the secondary cleft. But we can release a small amount of acetylcholine. We've got lots of surface area to catch and trap that acetylcholine on acetylcholine receptors on the sarcolemma. So action potential, action potential, action potential, all the way till we get to the end of the nerve terminal. And then we go from nerve to muscle using a physical signal. That physical signal is a neurotransmitter termed acetylcholine. Acetylcholine. Emma, I thought you had a question. Just on the side of my eye, I saw you raising it. Okay? All right. Are we okay with that? Nope. We've done this. We get acetylcholine. It binds to its receptor. And then it switches, and this acetylcholine receptor gets activated, and it generates an action potential on the surface of the sarcolemma. So we go electrical to physical, back to electrical. Okay? Why it does that, I have no idea. Okay? I have no idea whatsoever. But that's what happens. Okay? So this has happened. You've bound the acetylcholine. We now have to have a way to release the acetylcholine so you'll stop contracting your muscle. And that's done with an enzyme that is called acetylcholinesterase. acetylcholinesterase. There are lots of neurotoxins from fish and spiders and snakes and all of the things that I am terrified of that tend to affect one side or the other of the neuromuscular junction. Okay? They may block the release of acetylcholine, so you can't contract your muscles. That gets into your diaphragm and you suffocate and you die. Or they block the acetylcholinesterase and you can't turn off the muscle contraction. So your diaphragm seizes up, it cramps, eventually it fatigues, and you can't breathe. And you suffocate. Okay? That's how a lot of neurotoxins that don't cause tissue damage work, is they affect one end of this versus the other. Okay? So once we've found the acetylcholine, once we've generated an action potential on the surface of the muscle fiber, we need to move that signal from the surface of the fiber down into the inside so we can actually get actin and myosin to form a cross bridge and give us force. Okay? So it moves along the surface and then it goes down the T-tubule. And then the T-tubule tells the sarcoplasmic reticulum, the action potential here tells the sarcoplasmic reticulum to release calcium. That calcium binds to troponin, and then the troponin moves to tropomyosin, which allows actin and myosin cross bridge, and we get a contraction. This process, action potential to calcium release, is termed excitation, contraction, coupling. Okay. Excitation, contraction, coupling. Someone, you want to do something fun for me? You want to do something for me? Sure. You have books in your bag? No books? How heavy is your bag? Okay. Matthew, how heavy is your backpack? Not very. Anybody have books in your bag? I know you guys don't have books anymore. That's okay. Dylan, grab your bag. Matthew, may we borrow your bag for a moment? Okay, we're going to do a fun trick with Dylan. All right. Dylan, I want you to take the bags and I want you to hold them out like this. You do that? All the way out. Don't hit Madison in the head. Okay. While Dylan's doing this, Alicia, where does this contraction start? Good. Okay. Brain says, ooh, Dr. Black's making me contract. We're doing it, okay? Send action potential down the spinal cord. We're using our delts in a little bit to do all of this. We're going to get to about C5. We're going to depolarize motor neurons down those axons, across the neuromuscular junction, right? Action potential, calcium release, actin and myosin. Dylan, how you doing? Good. Good? Are you sure? Sure. Okay. This is where having the mask on becomes tricky. In the world of pain research, one of the things that we know is that you can lie to us about whether something hurts, but if we have a picture of your face while it's going on, you will make a very distinct, it's called the pain face. And you cannot, it's involuntary. So with this mask, I can't decide how it's doing. Okay? His arms are shaking a little bit. We are beginning to mess up right here in excitation contraction coupling. Okay? This little thing, remember we had to generate an action potential? Dylan, you're doing great. Hang in there, buddy. You're doing great. Generate an action potential. Okay? Sodium moves in, potassium moves out. We have a sodium-potassium pump to restore our gradients. Okay? As you do contractions for very long and you begin to fatigue and get tired, this cannot repolarize correctly and you cannot transmit a signal. Thank you, Dylan. You're a very good sport. Okay? And you get to a place where I can't release enough calcium to get enough cross bridges to maintain what I'm doing. Let's all clap for Dylan. Very nice. Now, no one will ever volunteer for anything that I do in class ever again. All right? All right. That's fatigue. That's why this is so unbelievably important. Right? That's why this whole process is so important. We need force to do things As we do them, though, as we exercise, things are going to change in the signaling area, and we're going to get them messed up, and then you can't get as much force, and you have to stop. Okay? I guess we could have just had him do reps to failure. That might have been an easier or a better thing. Okay? That's what happens, and that's why this process is so important. Impairments in excitation contraction coupling are one of the primary causes of fatigue, okay, that occurs outside of your brain. Okay? Dylan did a very nice job of hanging in there with us. Most people, when you stop exercising, you're not stopping because your muscles can't physically do anything anymore. You're stopping because your brain says, this sucks. Let's stop. Okay? When your brain says stop, that's called central fatigue. When your muscles say stop because this is messed up, that's called peripheral. Excitation contraction coupling is one of the primary sites of peripheral fatigue. Right? Peripheral fatigue. If you go work out and you do push-ups or sit-ups to failure, or you lift weights and you're really motivated, because I've had lots of pre-workout today. I'm super hyped up, right? And I got my beats on, and I got some great freaking music going. I'm gonna get this workout. Okay, you're really motivated. Oftentimes I did 5 reps, I tried for 6 and I couldn't get it, probably because of an impairment in excitation contraction. Okay? So there are lots of steps in going from brain to Crossbridge. Okay? But there are some of them that are more prone to getting messed up than others. And that determines why some people can lift more weights than others, or for longer, why some people can run faster than others, and things like that. Okay? So this entire pathway is really, really important. Any small breakdown anywhere along all of this, and the whole thing goes to shit. Right? And therefore, you have to stop running, or you have to stop lifting weights, or you have to go a little bit slower than what you wanted. Right? That's why I think all of this stuff is really, really important. Okay. So we've done the excitation contraction. We've formed actin and myosin cross bridges. They're there, they're ready, we're getting force production. Okay. Assuming what I had Dylan do was an isometric contraction, because he was holding them straight out, but assuming you're picking something like up, you're going to get a concentric or a shortening muscle contraction. Assuming that happens, you're going to initiate something called the sliding filament theory of a muscle contraction. And the sliding filament theory describes what goes on at the level of the sarcomere as myosin filaments pull on and slide actin filaments towards the middle of the sarcomere. Okay? And that force generation and that movement there is what leads to movements in other sort of the whole muscle. Okay? That's what's going to go on. So we bind, we have a cross bridge. The myosin head wants to tilt. That's called a power stroke. Right? That's called a power stroke. You get this power stroke, pulls the actin, and everything's good. Right? That's the sliding filament theory of contraction. Right? Within the sliding filament theory of contraction, we need to understand something that's called the crossbridge cycle. Right? So when a sarcomere moves, when those filaments slide, they move a tiny, tiny, tiny amount. Right? Because the amount they move is so small, you have to do that over and over and over and over again to generate enough shortening and enough movement to actually be noticeable and allow the whole muscle to move. And so to do that, we're going to have to do something called the crossbridge cycle. Okay? So note here at the start of what we're going to say is the start of the crossbridge cycle. Here's my actin filament. Here's my myosin filament. Okay? The crossbridge cycle describes what happens with one myosin head and its level of engagement with an actin filament. Okay. So imagine that we're showing one, but this is happening for all of your myosin. Okay. So when myosin goes from being unbound and then binds onto the actin filament, it is in what we call its high energy configuration. And by high energy configuration, that denotes that on the myosin head, there is attached a molecule of ADP and an inorganic phosphate molecule. Okay? The attachment of these two things comes from the breakdown of ATP down here later on in the step. But just trust me, it starts like this. Okay? It starts like this. So we form the cross bridge. We're in our high energy conformation. This happens, okay? And then the ADP and the inorganic phosphate are going to detach. They're going to detach, and the myosin head is going to tilt and try to pull the actin molecule, in this instance, from right to left. So it's pulling it this direction. So here's the head being straight up. Now here it is pointed and kind of leaning over to the left. Okay? So we've kicked out our ADP, we've kicked out our inorganic phosphate, and that's going to drive this power stroke. Okay? We've gotten a power stroke in step number 2. Please stay with me here. The next thing that happens to me is the most backwards thing in all of physiology in the known world. Okay? We're attached and we've done this. We're stuck. Okay? We've moved a tiny amount. We need to move farther. We're still getting lots of signal from the nervous system, lots of signal from the neuromuscular junction and from excitation contraction coupling, lots of calcium. We need to detach this myosin head from the actin. We need to break it off. Then we need to move it back to its high energy configuration so it can attach again, and we can perform another power stroke. Okay. That all seems relatively straightforward, except for how we detach it. We detach the myosin by binding ATP to it. ATP comes in, binds to myosin, it detaches. Okay? Then we're now, so it's in its low energy state. Then the myosin ATPase enzyme on the myosin head splits this ATP into ADP and inorganic phosphate, and then the myosin head's going to recock, and we're back in our high energy state. Okay? The reason this is backwards, you do not use energy to contract your muscle. You use energy to relax it after it has been contracted and then allow it to continue to do this. So you don't use energy to attach the myosin head or to generate the power stroke. You actually use energy to break it off and recock it so that it can go again. Right? That to me is like the most backwards thing. You go for a run or you're going to go this way, you're like, oh boy, I'm using lots of energy. You are, but you're using lots of energy because you're running the crossbridge cycle over and over and over and over again. You're breaking it apart. You're breaking the crossbridge a bunch. Okay? You're breaking it off a bunch. And that's where all the energy is coming. You guys want to guess how long did Dylan hold those bags out? Maybe a couple of minutes. Okay. If we ask Dylan to do this and to curl the bag or to take it from here and do this with the same muscle and do a shortening contraction, Dylan, you could do that for 2 minutes. I'm not going to ask you to. You don't have to.
Yeah.
Probably.
Yeah.
Maybe. It's light enough. Okay. I'm skeptical, but I'll believe you. Okay. So the type of contraction that we perform is going to have an influence on how many times the crossbridge cycle goes over and over again. That's going to have a role in how much energy it produces. Okay? So when you perform concentric muscle contraction, a shortening contraction, you have to do the crossbridge cycle many, many, many times. Okay? So it uses a lot of energy. When you perform an isometric contraction, hold, okay? Don't move, hold. No change in length, just right here. All you have to do is form the cross bridge and have it hang on. You don't have to do this because we don't need to shorten. We don't need the power stroke, right, to be broken. We don't have to go anywhere. So in general, when you do an isometric contraction, you use much less energy. So you should be able to, with the same muscle and the same weight, hold an isometric contraction a lot longer than you could do a concentric with the same weight and that same muscle. Even more remarkable are eccentric contractions. An eccentric or a lengthening contraction, again, it binds, you get a cross bridge, you get force, and then the external load is so great that rather than shortening, it actually gets pulled in the opposite direction. Okay? You guys think of a situation that would illustrate this point. Think of, if you're going to go run, let's just say, when does running, let's say you're going to run at the same speed. When or in what situation does running feel more difficult, and in what situation might it feel less difficult? Uphill versus downhill. Okay, very good. Riley, you want to guess? Have you ever run uphill and downhill, Riley?
Yeah.
Okay. You agree with Lauren? Uphill feels worse?
Yeah.
Predominantly, what kind of muscle contractions are you doing with your quads and your glutes to run uphill?
Isometric.
Right, good guess. There's part of it that's isometric. Up, you have to do both. I'm being a pain in the ass. Okay, but uphill, right, is more concentrically dominated than flat. You got to generate more force. What about downhill? Everybody agree running downhill feels easier than running uphill? Okay. When you run downhill, all you're really doing with your legs is an eccentric contraction in your quads and a little bit with your gastroc and your TA. All you're doing when you go downhill is you're just keeping yourself from falling on your face. Okay? I get to heel strike, and I'm going down, and I'm getting just enough force so that I don't go over forward. Okay? And then I just have to only move like the weight of my leg, and gravity sort of pulls me down. Okay? The reason running downhill feels easier than running on flat or level ground or uphill is because you do not have to run the cross-bridge cycle in those same muscles nearly as many times. If we measure your oxygen consumption and your energy use while running downhill, it is significantly less than running at the same speed on level ground and going uphill. Okay? So here again, we have structure and function, okay, on the level of the proteins in your muscles that are controlling metabolism, that are controlling how fast you can run, that are controlling your perception of how difficult that activity is going to be. So when I tell you guys that muscles are the single most important things in your body, this is why I think that. Right? Energy use during exercise is primarily from this. Okay? Caloric expenditure during exercise is primarily related to the number of times that you have to run the cross-bridge cycle and the amount of muscle that's activated to run the cross-bridge cycle during a workout. So when I say you don't burn very many calories lifting weights, it's because you don't do very many contractions. This doesn't have to happen nearly as often as when you're running or biking or elliptical. Okay? And that's why it feels that way. Like downhill feels less terrible compared to level ground, which feels less terrible than going uphill. It all comes back to this. Okay? We also use a lot of energy to repolarize the membranes and those kinds of things, but that's a separate issue. Okay. Questions about that? I will ask you to know this exactly as it is written on the test. Okay? You need to know the steps. You need to know their order. You need to know whether actin and myosin are bound. You need to know whether ADP or ATP or inorganic phosphate is there, and the high and the low energy configuration. This is so important. This is one of the few things I'll ask you just to memorize, right? Because it influences everything else. Everything else. Okay, so we did this, blah, 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 blah. Already talked about that. Okay. So, I did this really well. To sum up, okay, in our last few minutes here, the idea of cognition to contraction, from brain out to Crossbridge, okay? Motor cortex decides we want to do some sort of a movement, okay? For many of you all right now, you may want to flip me off and be like, can we please, for the love of God, go, Dr. Stop talking, okay? Motor cortex, it communicates with an action potential. That action potential moves down the spinal cord, continues to be an action potential, moves all the way down to a motor neuron, okay? Again, action potential. And the motor neuron depolarizes and generates its own action potential. We call that motor unit recruitment. That motor neuron gets turned on. That's called recruitment. We'll talk about that on Monday. Okay? Action potential moves down the axon to the neuromuscular junction. We release acetylcholine into the junction. It binds to its receptor on the sarcolemma. And the sarcolemma generates its own action. Okay? That moves along the sarcolemma across that membrane, and it gets down into the T-tubule. It tells calcium to be released from the sarcoplasmic reticulum. Calcium binds to troponin. We get a conformational change. It moves tropomyosin out of the binding site. We then form a cross bridge where actin and myosin are going to be together. We get the cross bridge cycle. We generate force. Maybe we generate movement. We'll talk about the hows and whys of that on Monday. Okay. And then the whole thing starts all over again. While this is happening, the motor cortex, this all takes fractions of a second. The motor cortex is continuing to send more and more and more signals so that this continues to happen. Okay. All right. Thank you, guys. There is no assignment for Monday. You guys have a good weekend. Be safe. Make good choices. Wear a mask. Don't drink and drive. I don't know. Use the buddy system. Don't get in a car with people you don't know. I don't know. Oh yeah, shit, that's right. I will not see you all on Monday. I will see you all next Wednesday. Enjoy your long weekend. No, wait. Monday's Labor Day.